automated platform, the Tigris system. So this really allows them to test a lot of samples in one afternoon. And it's based on a duplex assay that targets conserved sequences in 2 separate regions of the XMRV genome. And again, they found that none of the samples in the phase 2b panel were positive at any time point. There's one caveat to this in that their assay has not been fully validated for other MLV-related viruses, only for XMRV, and so it's possible that there was a lack of diversity in this assay, although based on the sequence alignment, it doesn't look like it really should be limited. However, they are developing a second-generation assay that should detect generic MRV viruses. So moving on to the WPI results, they performed the same nested RT-PCR. As I meant to point out but forgot, they have yet to process the whole blood samples, and so therefore we're not reporting the results from any of the other laboratories in terms of the whole blood, only for the PBMCs and the plasma, and those whole blood samples have yet to be decoded. So when they looked at PBMCs, however, they found that none of the, when they looked at the plasma samples, they found that none of the samples from any of the subjects were positive. So we're not quite sure why the plasma is negative for all 4 of the labs, even the 2 labs which were able to detect it in the first place. Whether this was a processing issue or there's just a natural variation in the levels of virus in the plasma is one possibility. However, in every instance, all the labs found the plasma samples to be negative. The WPI were able to detect positive samples in the PBMC samples from 3 out of the 4 subjects. However, they also found 1 out of the 2 time points in the pedigree negative also to be positive. Investigation following decoding of the results determined that there was a procedural error during the extraction of nucleic acid from these PBMCs, which likely introduced cross-contamination of the samples. So really we can't draw any conclusions from any of the positive results in the PBMCs from this study, unfortunately, including the time points and the different subjects, because really there was probably potential contamination of all the samples. So this is just showing a summary of the phase 2 serology results, which we added to the study after the phase 2A study, just to give some more information about these subjects and detection of virus in them. So the CDC performed, only the CDC and Frank Rossetti at the NCI currently have serology assays, so only these 2 labs participated in serology at this time. The CDC uses a Western blot for multiple MLV antigens, so this involves taking generic mouse leukemia viruses and running them out on the gel and then blotting that with the patient's sera. And in this instance, all 5 of the subjects were negative for any XMRV antibodies by Western blot. Frank Rossetti at NCI uses a flow cytometry-based assay very similar to the assay described in the Lombardi et al. Paper published last year, which involves expressing the envelope from another mouse leukemia virus, spleen focus-forming virus in cells, and then detecting anti-XMRV antibodies with that envelope. In this instance, he did find 3 of the 4 subjects to be seropositive. One of the subjects was only seropositive on day 0, but not day 2, which is again strange that antibody levels really shouldn't vary in the plasma, even if it has a delay in processing. 2 of the other subjects were positive on both day time points, and however they also again found a false positive with the pedigree negative, who was positive on the day 2 time point, but again strangely not positive on the day 0, suggesting that this may be true false positivity. So this is just a summary of all of the nucleic acid serology results for this phase 2B study. So again, to emphasize that 3 out of the 4 labs, including the CDC that had found samples positive in the first phase, found them completely negative both by nucleic acid and serology. The WPI did find PBMCs positive, but not plasma, and then all of these, 3 of these 4 subjects were positive by serology, but there was also a false positive, and there's no real correlation between which time points were positive by serology and which were positive by nucleic acid testing. So in conclusion, in this round of testing, only 1 out of 4 labs detected XMRV in clinical samples, and only in the PBMCs. Ultracentrifugation by the NCI DPR lab or direct extraction of plasma did not seem to make a difference in detection, and sensitive nucleic acid testing from a diagnostic company was unable to detect XMRV. So just a couple of notes on this phase 2 pilot. As I said, it was really designed to validate sample type and also whether our national repositories would be candidates for extensive testing. It really wasn't designed or performed as a clinical evaluation or a longitudinal study. It's really impossible to draw any conclusions from this extremely small study, particularly as there was a loss in plasma positivity for all the samples from all of the labs testing, and there was false positivity in PCR due to processing issues, and there was false positivity in the serology. So in conclusion, really based on both phase 2A and phase 2B, we don't really see any clear advantage to delayed processing. If we only looked at phase 2A, there could be an advantage, but we really don't feel that there's sufficient evidence to go forward with that. And however, rather than really repeating this phase again and just going around in circles, we felt it was important to try and continue on to phase 3 and include more positive and negative samples and really get an idea of the sensitivity and specificity of these assays. So we're going to revert to standard next day processing. And while this is somewhat of a unhappy compromise, the labs that have found positivity in samples have done it using samples which were processed in a standard manner, so we're fairly confident that we're not going to lose too much in terms of sensitivity. And then I think it also highlights that serology is equally important as nucleic acid testing, and we should include it in more detail in future studies, and we want to bring on board into the study some more laboratories, particularly some of the commercial companies that have serology testing and can perform it in a high throughput manner. So this is just some of the initial acknowledgements, the people in the labs that actually did the work. I won't go through everybody's names. And then finally, there have been some comments and suggestions that maybe this work hasn't been performed as fast as it could be. And while we agreed that there were steps where it could have been performed faster, we've just put this timeline together, which is, we just put it together last night, so it's a bit rough and incomplete. But it really demonstrates from the very first publication in October how fast we have been able to pull these studies together, and kind of highlights the gaps that occur between the panels being distributed to labs and when those labs report back results, just because these studies are very low term through, and a lot of the samples we wish to be verified by viral culture, which, as I said, can take up to 42 days to get a positive result. So therefore, there's clear gaps, unfortunately, in the time with which these studies can be performed. So we're currently here at the BPAC last week. We basically reported all the results that we currently have. So we're now waiting for the collection of the samples for phase 3 to be initiated, which hopefully should occur in January, and then these samples will quickly be distributed to the participating laboratories. And one final additional point to point out is that some of these delays are also introduced because in order to collect human samples, you do need proper human subjects approval, and getting that approval also takes a couple of months. So that's one delay that unfortunately ourselves and Judy Mikovits at the WPI we can't control for. So with that, I'd like to hand the talk back over to Mike, who will go through some of the future studies that we had to perform.
Thanks, Graham. Just try to pull up presentation. Good. Okay, I think I'm ready. Kim and Graham, can you see my presentation?
Yeah, I can see it.
Great. Okay, so yeah, this is a presentation that I did give following Graham's at the BPAC meeting, and it's really a longer-term planning sort of review. In that, as we're doing these studies and trying to identify and establish the performance of these assays, and particularly higher throughput assays that can be applied to much larger numbers of samples to understand the prevalence and transmissibility of these viruses in the blood donor recipient populations, we need to be moving ahead of the process in terms of getting funding and approvals to access panels and launch studies. So what I'm going to present is a brief summary of what are currently proposed and submitted grant applications to NIH to support both retrospective and potentially prospective studies. So the first slide here is just outlining the scope of the proposal. This involved a study that would exploit the repositories that the National Heart, Lung, and Blood Institute has had the prescience to fund building over the last 40+ years. And the original repositories were designed to study hepatitis or HIV or other emerging infections, but they, again, have had the foresight to maintain these frozen repositories in a national central archive. And we've been fortunate to be involved in building most of these repositories, and are very familiar with their content and the ability of these repositories to be accessed for purposes of understanding XMRV, MRV prevalence and infectivity. So in looking at the range of repositories, there's dozens of these, both in the blood banking and other aspects of research relevant to National Heart, Lung, and Blood Institute, but there were 5 that really we felt were particularly valuable to study. In particular, the first 5 that are listed here could be used to establish the prevalence of these viruses in the donor population over the last 4 decades. And then 3 of them had linked donor recipient samples that would allow us to look at the rates of transmission and the correlates of transmission, meaning is transmission associated with donations that have nucleic acid positivity, antibody positivity, and are different recipient groups more or less susceptible, and also is the way the blood is processed in terms of removing white cells, which is now standard practice in most blood banks in the U.S. by filtering blood, a process we call leukoreduction. Is that effective at preventing transmission of XMRV viruses? It is effective at blocking transmission of other cell-associated viruses like cytomegalovirus and human T-cell leukemia virus, HTLV, another retrovirus. So if viruses are predominantly cell-associated, then filtration can remove the cells and allow the getting a flash screen here. Sorry. And then another variable in blood transfusion medicine is that the storage of blood. The blood can be transfused within several days of collection or more than a month after collection, and that can influence the infectivity of positive units. So we have that information on these repositories, so we can look at that question. And then again, for these 3 repositories, the transfusion-transmitted viruses, the viral activation by transfusion study, and what we call radar repository, we do have outcome data from these recipients that gives us some information on mortality and morbidity that would allow us to see whether in the early period following transfusion, if transmission is occurring, do we see evidence of disease in the exposed recipients, infected recipients, compared to the uninfected controls. Now, for these studies, we've been encouraging and facilitating, to the extent we can, partnerships with the large manufacturers that support blood banking, including companies like Abbott and GenProbe, in great part because we really need the throughput. As Graham indicated, the labs that are participating in the current, government or the WPI or NCI labs, their technologies are labor intensive, and so we send a panel of coded samples, perhaps 50 specimens, and it takes several months for results to be generated. We're now going to be talking about testing tens of thousands of samples, and if we have to screen the blood supply, we're literally talking about tens of thousands of samples every day being donated and tested. So we need to work with these manufacturers to build serological and nucleic acid tests that are compatible with this kind of throughput. And we also need these tests, should donor screening be warranted, but we really need that kind of throughput and performance. GMP, as we say, manufactured reagents on very automated and validated platforms to really execute these retrospective studies as well. So this next slide really just outlines the content of these 5 repositories. I'm not going to go into the detail other than to point out that, again, the earliest repositories here were collected back in the 1970s. The most recent in the early 2000s. So they do span 4 decades and will allow us to assess whether this virus has expanded in the U.S. in the donor population or is really been a virus that's been around at stable levels for decades, which influences our perspective on the relevance of the virus to blood safety, because at this point there's not a lot of epidemiological evidence that transfusions are associated with clinical CFS, but those studies are small. So we think that time element is important. Then I've highlighted in bold here that the 3 repositories that have linked donor recipient specimens, and these repositories literally have thousands of donations that were transfused into recipients for whom we have pre-transfusion and in most cases multiple serial post-transfusion specimens. In the more recent time period, we have both plasma and frozen whole blood, so we're able to look at both the prevalence of the virus in the donors and the recipients in cellular compartments of blood, the PBMC type of sample Graham summarized and has been used in many studies to detect XMRV or the other polytropic MLVs. So this is an important additional piece here. Also, just to mention that most of these studies were done in fairly standard transfusion recipient populations, elective surgery, some other subgroups. But the one that's called the VATS, the viral activation by transfusion study, was actually focused exclusively on HIV-infected patients, mostly with fairly advanced immunodeficiency and AIDS. And so that gives us the opportunity to look at susceptibility and disease outcome if transmission is observed in an immunosuppressed population. Also, both the VATS and the last repository we call RADAR were studies that included half the donations were filtered to remove the white cells and half not filtered. So we're able to also examine the effect of blood filtration on transmission, as I mentioned earlier. So just to touch on the aims and sort of the projected findings from these studies once executed, the first aim really, as I alluded to, is to really establish the prevalence of these viruses and the demographic correlates of infection in U.S. blood donors. So the concept was to take those 5 repositories that were on the prior slide and just take a representative group of 2,000 specimens from each repository, both plasma and, if available, whole blood, and perform nucleic acid testing and antibody testing. And again, given the numbers we're talking about, we're going to need to employ tests on automated systems, and so we've developed formal commitments and agreements with GenProbe and Abbott to supply the tests and or perform the testing to the samples. Now, if we just take the data from prevalent studies that have observed these viruses in the donor healthy control group or donor populations, that they've ranged from 0.2 to 7%. So given the 10,000 donations that we would be studying, we would potentially detect 20 to up to 700 positive donors, either with nucleic acid positivity indicating viremia or antibody. So we could, if we do detect virus consistently, then we can look and with reasonable numbers in the 50s, 100s, we'll be able to really then look temporally across those 5 repositories to see, has the prevalence been stable or is it increasing? Look at the relationship between gender and age of the donors, racial, ethnic groups, perhaps geographic regions, and see whether there's any evidence of sort of epidemiological associations with these demographic parameters. The second and certainly probably most important aim, assuming that we demonstrate prevalence in aim 1, we would then move on to the most precious samples in these repositories, which are the linked donor-recipient subsets of specimens. And this would then allow us to address the question of transfusion transmission rates and, again, correlates of transmission. So, again, the correlates I'm referring to are the detectability of the virus in plasma or cells and the presence of antibodies. And clearly, that's going to be critical information to correlate with transmission to guide a decision as to whether screening would be nucleic acid-based screening, such as we now do for West Nile virus and HIV and hepatitis viruses, or antibody-based screening, which is done for a larger array of infectious diseases, or both. And so really this is the critical information we'll need to guide strategies towards donor screening. As I mentioned several times, the impact of filtration, removal of the white cells on transmission, as well as the age of storage of blood on infectivity, and then these effects of underlying disease or immune status of the recipients on acquisition risk. So particularly comparing relatively healthy elective surgical recipients with these HIV-infected, predominantly end-stage AIDS patients who were transfused in that batch repository. And again, just in terms of numbers, we in these repositories will have what we call 11,000 quote donor exposures, meaning that we'll have recipient pre- and post-transfusion samples for recipients who've received 11,000 different blood transfused components. So if we do have prevalence rates in the 0.2 to 7% range and transmission rates in the 20 to 50% range, we'll identify a fairly large number of transmissions. Certainly several hundred potentially transfused XMRV, MLV positive units. So this will allow us, if if the assays all perform well and the virus is prevalent in the donor pool, to really address critical issues on transmission risk and determinants or correlates of transmission. And then finally, the third aim is to really try to get information on the course of infection and the clinical outcome in recipients. So if we do identify recipients in the aim 2, as I alluded to, in many of these repositories we have longitudinal samples, serial samples collected days to weeks to months following the transfusion. So we would study those to characterize the levels of virus, the diversity of the virus, and compare the virus in the donor with the virus that's transmitted to recipients, attempt to perform cultures, and do careful antibody characterization to document the rate of seroconversion and the antigens that are inducing antibodies to guide further enhancements of performance of screening serological tests. We'll also have some data on the survival and clinical findings from these recipients, not all the data that we'd like if we were doing a prospective study, but sufficient data to at least examine whether we do have higher rates of death if transmission is seen in some recipients compared to the non-infected recipients, and if some critical symptoms that we did capture data on are increased in the exposed recipients. So when we think this data, if we're successful at capturing perhaps dozens of recipients with serial samples, will really help inform the understanding of the acute evolution of the virus and the antibody response, and that this will help us in terms of blood screening, but also I think guide further insights into the pathogenesis of the infection in general. So finally, just to mention that that although it's too early to really begin to execute, and we don't have the tests yet that are validated from companies. We are beginning to think and discuss with FDA and the company partners the potential of prospective screening. So again, and thinking about the funding to support such large-scale studies. So this next slide really just explains that that process has begun, even though we're not sure whether this will be warranted or when it would be feasible. We are considering the potential need for prospective trials that could lead to FDA approval of donor screening assays, recognizing that those trials would probably involve both NAT testing and antibody testing. Once we would start these kinds of trials, ethically we'd probably be doing these in the context of real-time screening, meaning that the donations that are reactive would not be transfused, but would rather be discarded or further studied. We would be also enrolling those positive donors into follow-up studies with controls, both positive CFS patient controls as well as negative control donors, to study immunologic and other findings to help, again, in terms of pathogenesis research. And then we'd also do what we call look back, because our assumption is that these donors who we detect probably have been infected for quite a while. So we would trace recipients of prior donations to determine if they got infected, further informing that question of transmissibility by blood transfusions. And again, it's a debate within the industry and with FDA as to when such studies would be warranted. There are other studies going on now. Dr. Alter is conducting some studies, and the Red Cross, Drs. Kramer and Dodd, are beginning to design and consider moving forward with some cross-sectional studies, as well as some repository studies. So this is not the only work going on. I think we're probably at least 6 months away from completing the phase 3 study and 4 study and getting data from these other studies before we would be prepared, and the companies would be prepared to embark on IND trial. So with that, I'll pass it back to Dr. Kleinman to lead the questions.
Okay, well, thank you to both of the speakers, and I note that we are running a little bit over time. It's about 13 minutes past the hour. We do have, I think both the speakers have agreed to stay on till half past the hour, so we'll use the remaining portion of the time to have a question and answer session. We're covering some questions submitted in advance by Thursday afternoon. A number of these questions that were submitted, answers have already been included in the presentation, so I will be selective in the questions that I ask. But before doing this, because the Blood Products Advisory Committee considered an important donor question at its meeting on Tuesday, I wanted to update the webinar participants on what happened on that particular issue. FDA had posed the following question to the BPAC committee for a vote. The question was, do the scientific data support asking donors about a medical history and or a diagnosis of CFS as a basis for indefinite deferral? That is, those persons would not be allowed to give blood either now or in the future. It's important to note that the question was not about whether the BPAC thought that patients with CFS should be allowed to give blood. Rather, the question addressed a very specific issue, and that is whether a question should be added to the donor history interview questionnaire, and that question would inquire about a medical history or diagnosis of CFS. That is as opposed to using procedures that are currently in place that have recently been implemented at most blood centers. And most blood centers now are providing educational materials to all people who come to donate, and educational materials about CFS, and asking prospective donors to let the blood center personnel know if they had a medical diagnosis of CFS and consequently not to donate. Another important issue is that the discussion amongst the BPAC and the speakers prior to the vote considered the issue of donation with regard to impact on the health of the donor, with regard to protecting recipients of blood donation against recipients of blood against acquiring XMRV or MLV infection from the donor's blood, and also whether recipients could acquire other viral infections if they were transfused with blood from persons who had CSF. CFS. So the vote here, here's the question again, and the outcome was 9 of the members voted yes and 4 voted no. The vote reflected, again, the opinion on the issue of whether asking a question was better than using educational materials to elicit the donor disclosure. All BPAC members actually indicated that they agree with the indefinite deferral of CFS patients based on all the evidence with regard to both donor and recipient safety. So that's a brief summary of the vote. I want to now go on to the questions and answers. The first question will be actually for both speakers, but primarily for Dr. Simmons. There are many terms being used to refer to these agents. So we know about XMRV. We've also heard MLV and MRV in the presentations given today. Other terminologies that we've heard is HMRV for human MRV or HGRV. And the question was, do these mean the same thing or different things? And what really is the best terminology that we should use right now? So Graham, would you like to answer that question?
Okay, so this is obviously a difficult question to answer. Unfortunately, retrovirology in general and MLV in particular, naming has always been a minefield with lots of many different names out there for basically the same thing. There is a difference between XMRV has clearly been isolated, and this full-length infectious virus has been cloned and is named to exist, whereas the other viruses that were isolated by Dr. Lowe, which they called this MLV-related viruses or MRV, unfortunately so far full-length sequence and infectious virus has not yet been identified. So it's important to separate those two sets of viruses at the moment into XMRV and MRV, with XMRV really being the only one that we really know exists as a fully infectious virus at this time point. Other than that, and then MLV in general, mouse leukemia virus, is kind of a generic name for the whole family and can include these viruses that only infect mice, endogenous viruses, exogenous viruses, etc. And some of the studies are using samples and primers, etc., which are based on MLV. So unfortunately, we do need to continue to use that term. The other terms, HMRV and HGRV, which I believe stands for human gamma retrovirus, have really been introduced to try and differentiate these viruses from MLVs and demonstrate that they really are human viruses, and probably at this time they're not called for, and later on when the virus has really been further characterized, I think probably someone will sit down and come out with a unified name.
Okay, well thank you. Let's go on to the next question, which is what is meant by pedigreed positive and negative samples? And based on the results of the phase 2 study, do we actually have pedigreed positive and negative samples for XMRV and MLV in CFS patients? Do those samples exist?
So really the pedigreed sample, the way we're using the term is meaning that multiple laboratories and multiple assays have found the same result, that it's either XMRV positive or negative. At the moment, unfortunately, not enough of these pedigreed samples really do exist. Again, as I said, viral culture is probably the gold standard, and again, that's taking 40-odd days to perform. So doing that on a large number of samples is proving difficult. So yeah, currently we probably need more pedigreed samples, and we really need to build up a sufficient panel, and I believe the FDA are also looking into building panels of positives and negatives.
Okay, well, next question. Based on the phase 2 results that you described today, has the issue about the ability of different laboratories to identify XMRV or MLV in clinical samples been resolved?
No. It looked like we were getting there with the phase 2A samples, but unfortunately with the phase 2B, I don't think so. And so again, we're really going to have to wait to the phase 3 where we have a lot more positives and a lot of negatives to really answer that question.
Okay, the next several questions cover some of the things I think that Graham, you've already addressed, but perhaps these will serve to emphasize some of the points. What conclusions can be drawn from the differing results obtained in phase 2A as compared to phase 2B?
At this time, I'm not sure too many conclusions can really be drawn. And again, I think we have to wait for phase 3 to really understand in more detail. As I said, we are going to investigate the delayed processing aspect in a separate study with Judy, but at this time we're just going to revert to standard testing.
Right, right. Another question, what are the possible explanations for the differences in findings between the laboratories in phase 2B specifically?
The different assays which have been employed, that maybe there are subtle sequence variations in these viruses in the clinical isolates that some of the assays just aren't picking up. Obviously, unfortunately, contamination is always still a potential. That's really the two explanations we're working on at the moment, and that maybe there's just a real variation in the levels of virus in an individual over time. And this is seen with particularly hepatitis C, but also HIV to some extent. Viral loads do vary quite dramatically sometimes, and it may just be that we're right on the limit of detection, and sometimes it falls below the level, sometimes it's just above the level for some of the assays.
Okay, and then the final question on this aspect on phase 2 is on the actual studies is what does it mean that there were false positives? And by definition, these are positive results in samples where we expect to have negative results. What does it mean that there were false positives in phase 1? We saw that from 3 of the laboratories, and in phase 2, which we saw from a couple of the laboratories.
Right, the phase 1 I think we're not worrying too much about because they were explained by subsequent sequencing of product and found just to be a non-specific genome product, and all the laboratories which do employ nested PCR are using sequence confirmation, so really that's not too big an issue, I don't think. In terms of the phase 2, again, we have an explanation that's hopefully a one-off error in the processing of the samples, and hopefully, well, certainly unfortunate, shouldn't directly influence any of the future results or previous results.
Okay, thank you, Graham. I have a few questions here that I think would be primarily addressed to Dr. Bush. Mike, the first question is concerning possible transfusion transmission. You've addressed how this would be studied, but the first question is there any evidence currently that this virus has been transmitted by these viruses, or the virus group has been transmitted by transfusion?
Yeah, thanks, Steve. There really isn't. There have been a small number of published studies that have included case control studies of CFS patients that have included questions regarding transfusions, and those published studies have seen no correlation between receipt of prior blood transfusions and subsequent development of chronic fatigue syndrome. There was also a presentation at the XMRV workshop that was also summarized at the recent BPAC meeting, a recent survey that primarily was focused on a history of donation by patients who've developed CFS, but also inquired about history of prior transfusion before or subsequent to diagnosis of CFS, and that really found rates of transfusion that are essentially identical to transfusion rates in the general population. There are certainly, been some anecdotal descriptions of patients who have developed this syndrome following transfusion, but again, in terms of systematic studies, there's really at this point, no evidence of transfusion association as a major contributor to CFS. That doesn't rule out transfusion as a potential vector or transmitting mechanism, but it's unlikely to be a major, significant contributor to this disease.
Okay, and next question is, why can't tests currently being used by VIP Diagnostics and WPI for XMRV detection be used to test the blood supply? Why can't such tests be used?
Right, and I think both Graham and I, kind of emphasized that the current testing that's being performed in all of the labs, with the exception of GenProbe, for the virus, especially the nucleic acid test, but also the serology, simply don't have the throughput and the turnaround time that's absolutely critical for blood transfusions. Blood is collected and literally tested that same night. Platelet transfusions can only be given within 5 days of collection. Red cells, 6 weeks, but that testing must be turned around quickly as blood is critical and always short medical therapy. So we really need to move these tests onto highly automated testing platforms. Not only the instruments are much more automated and avoid concerns around contamination and have high throughput, but the reagents also, the tests themselves are manufactured and approved by FDA with extremely reproducible and high quality and high volume production and performance. So this is really why we've been working hard, and the companies that make blood screening tests, I think, now working hard to develop assays and allow the studies to go forward and with the mindset that eventually screening, if indicated, could be implemented as it is currently for all the other viruses with same day essentially testing.
And next question, as we're winding up towards the end here, that what hasn't been addressed, I think, on this call is what about tests to determine actual viral load of XMRV and MLV, so quantitative tests? What is the status of those tests?
Yeah, the assays that several of the assays.
Well, go to webinar, web events made easy.
The kinds of tests that are used for viral load quantitation for HIV and hepatitis and other viruses, and certainly the results from the analytic panel and results that I've seen from the companies are highly indicative that quantitative assays will be fairly readily developed as diagnostic patient monitoring tools. I've seen data from GenProbe and Abbott, for example, and the real-time data from several of the other labs involved, the more academic or government labs, that clearly indicate that quantitation is not going to be a problem. I think the issue is that the viral load is so extraordinarily low, which is suggested by a number of the findings we've been getting. Quantitation may not be as much as an issue as simple detection and adequacy of sensitivity. Perhaps people will have to do replicate assays of maximal input on individual patients to even consistently detect the infection. So, but in any event, the technologies and the data we're seeing clearly indicate that viral load tests are probably in the horizon near term.
And I think your answer sort of touches on the very last question I'm going to ask, and that is there currently a reliable test for XMRV or MLV? So I think we've been talking about that issue throughout the webinar, but if you'd like to take either one of you a kind of final concluding statement about is there a reliable test for XMRV and MLV?
I think the term reliable is, it needs some sort of definition here, and I think these tests that are being employed in experienced, reputable labs and certainly on the analytic performance look to be quite good. And I think the question that we're facing is, are there any tests at this point that have been proven to reproducibly and, if you will, reliably or consistently detect this virus or antibodies to this virus in clinical samples? And I think there's still work to be done. I'm really optimistic the phase 3 panel that's been designed and is about to be compiled will really more comprehensively determine the reliability and consistency of detection in a much larger group of patients versus controls. So I don't think we're at the point of having any tests that have demonstrated clinical performance to really ascribe a high level of confidence that we can detect this infection in patients with good sensitivity and specificity.
Okay, well, thank you, Mike, and thank you, Graham. That winds up the question and answer session, and I'll just turn it back to Kim for closing remarks.
Okay. Can you hear me, Steve?
Yes.
Okay, I've had some problems with my mic and other things throughout the webinar. So just to offer thanks to all of our participants today and Dr. Simmons, Dr. Bush, and Dr. Kleinman. I can tell you that they have spent hours preparing for this presentation in addition to the ones that they gave on Tuesday, and was receiving emails from them at midnight last night and 6:27 a.m. this morning, so a lot of thought and preparation has gone into making this material available, and we greatly appreciate it. Just a few resources, and we'll include these in the follow-up email for more information, specifically the webinar recordings from the earlier programs on XMRV. There were some pretty basic questions that came in that might be answered by some of the other materials that we have listed here. And just finally, for those who aren't familiar with the CFIDS Association, a statement of our mission, strategy, and core values. This is, I believe, our last webinar of 2010, although I said that on December 9th when I thought I was giving the last webinar, and we were able to pull this one together with the cooperation of the Blood Working Group and our speakers today. So we'll be putting out information about a schedule for 2011 and hope to continue providing the community with this kind of very timely and informative material that's not otherwise widely available. So with that, I'll just conclude, and thanks again to our speakers and to the audience and to all those who support the association and its work. Have a good afternoon, and look for the email you'll receive in a day or two with the follow-up information. Thanks so much, everyone.